Okay, everything seems to be good. We are just getting over 100 participants now, which is nice. So we will give it a minute and then we can start. Can you just confirm that you hear me loud and clear?
Yes.
Okay, perfect. Thank you so much. Fantastic. So I think we can slowly start because we just hit the 1 minute after 4 p.m. So hello, everybody. Welcome to our EMN live webinars. I would like to welcome all the attendees. Good morning. If you are from the North or Southern America, good afternoon. If you are from Europe, or good evening or night from India or Asia. So welcome to our 11th session at our EMN webinar. Today it's hosted by me, Aleš Kvasnička. I'm a PhD student from Faculty of Medicine, Palacký University, Olomouc. I will be your host, and my co-host is Daniel Mutithu from University of Cape Town from South Africa. And we are both EMN committee members of the Metabolomics Society. We would just like to give you a quick shout out to our social media where you can connect with us. And that's for our EMN network, but also the Metabolomics Society. We are active on Twitter and LinkedIn, but also you can email us for any questions. So I would just like to highlight one more thing, and that's where you can get your voice highlighted a little bit more and your presence. And this is our initiative of the Early Career Members Network, which is called ECR Voices, so Early Career Voices, where we are trying to highlight young scientists from metabolomics field. And we are already proceeding with this. Maybe if you are following us on social networks, you have seen some of these highlights where we always put like a short bio of early career scientists from the metabolomics field and share it through our networks for better reach. If you want to participate and get this beautiful, nice bio shared by Metabolomics Society of you and your research, you can just email us or reach us by direct message in Twitter or LinkedIn. And that's all from me. With no further ado, we would like to welcome Professor Gary Petty, and I will transfer my words to Daniel.
Thank you. In today's webinar, we are pleased to host Professor Gary Petty. He is the Michael and Turner Powell Professor at Washington University in St. Louis. Professor Petty is a pioneering figure in metabolism and metabolomics and has trained many researchers globally. He is renowned for developing award-winning gold standard softwares for processing metabolomics data. He has significantly contributed to the explanation of metabolism rewiring in cancer, showing lactate as a primary fuel source for tumor. His collaborations with leading instrumentation companies such as Thermo Fisher and Agilent has facilitated his contribution in technological advancement in metabolomics. Professor Petty has also established an NIH Multiomics Production Center at Washington University that focuses on collecting extensive genomics, proteomics, and metabolomics data for the multi-omics of health and disease consortium. Today, we'll be talking about the forward and reverse approaches to study human metabolism. Please submit your questions using the Q&A tab during or at the end of the presentation, and then we shall read out your questions during the Q&A session at the end of the talk. So with that said, it is my great pleasure to welcome you, Professor Petty, and the floor is yours. Welcome.
Thank you very much. Share my screen here. Can you see the screen okay? Confirm audio and screen sharing?
Everything
yes.
seems to be perfect.
Perfect. Thank you very much. Well, thank you very much for that kind introduction and the wonderful invitation to participate in this excellent forum. It's really a pleasure to be here. And thanks everyone for tuning in from all over the world, no matter what time of the day it was, is highlighted, sometimes more convenient than others. But thank you so much for tuning in. As noted, what I'm going to talk about today is forward and reverse approaches to study human metabolism. And what I mean by that is that if we're interested in studying human health or human disease, there's really two different ways that we can do that using metabolomics profiling. And the talk today is going to address each of those different approaches. So in one sense, which I'll refer to as the forward approach, you can start with human populations themselves. Often when we do that, we like to start with large populations, and we'll walk through exactly how we approach that in my lab. And then the second way you can do it is the reverse approach, where you might start with a model system of a particular disease, perform metabolomics profiling, hopefully come up with some interesting and exciting observations, and then you work your way backwards and ultimately validate in humans. So whether you're going from starting with humans for observations and then doing mechanism and model systems, or starting with model systems and working your way up to humans, really the difference in what I'm going to be talking about. So we'll start here with the first topic, and that is big populations. And before I mention that, get into those details, what I'd like to do is provide a little bit of a slide here on background nomenclature. So the way that we think about the molecular universe in our lab is shown here, with large molecules on the left and small molecules on the right. The large molecules, of course, are going to be things like your genetic material, your DNA, RNA, and proteins. And there are a lot of excellent technologies out there to measure those molecules. It's not going to be the focus of today's talk. But then on the right, you have the small molecules. And in that bin, I would place metabolites, but also lipids, so hydrophobic small molecules, and additionally chemical exposures. And I'm going to lump all of that for the purposes of today's talk. I'm going to lump profiling all of those molecules under the term metabolomics. Now, sometimes people will call that metabolomics, lipidomics, and exposomics, but just for simplicity, so we don't have to carry all those cumbersome words around all day, I'm going to use the word just metabolomics to refer to the profiling of all of those different types of chemicals. And what I'm going to start with, as I said, is measuring, starting with human populations, profiling there, hopefully ending up with some interesting observations that you can then try to understand from a mechanistic perspective by going into model systems as appropriate. And to do this kind of work, it's really critical, if you're going to start with the human populations, to be able to have large cohorts. And that's not actually trivial for metabolomics. If you think about the early days of metabolomics in the mid to late 2000s, when a lot of the metabolomics technologies that we all use today were first coming online, those approaches were, for the most part, optimized, benchmarked, and developed for relatively small numbers of samples. So I've shown you that in the top left-hand side of the slide here. Maybe 10, 20, 50 samples. But as metabolomics has matured, as much like in the spirit of many of the other omics, there's been greater interest, and there's been the draw to try to do larger numbers of cohorts. And so in 2024, you flash forward 20 years, and we're trying to profile cohorts with untargeted metabolomics of 20,000. And so what I'd like to just talk about today is, going from a small scale study, maybe n equals 10, n equals 20, all the way to these much larger cohorts that you might do in the spirit of some of the other genomics types analyses that have been done for other kinds of studies. And it's important to point out why you want to do this. And one of the critical pieces is the diversity of the population. There's an increasing appreciation for how diverse the population is, and how each of those how that diversity influences our biology. And there's this paradigm of precision medicine or personalized medicine, the idea being that we really need to profile and capture all of that diversity if we're going to understand how people have different kinds of diseases, how their diseases might respond differently to specific kinds of therapeutics, and et cetera. We really need to appreciate that. And so that's really one of the motivations for trying to do these larger scale studies. For the purposes of today's discussion, I'm only going to focus on performing untargeted metabolomics by using liquid chromatography mass spectrometry, here abbreviated LCMS. And I'm going to divide the untargeted metabolomics LCMS-based workflow into two simple steps. Now, obviously, this is a simplification, but I'm going to divide it into sample preparation and performing the LCMS, and then processing of all of those data. As you'll note, I have two traffic lights on the slide on the left-hand side of the slide. The top traffic light is green and the bottom traffic light is red. And that is to indicate that scaling up sample preparation in LCMS is relatively straightforward. There's a lot of automated sample handlers that allow us to do sample prep at large scale. And LCMS, although maybe not for untargeted metabolomics, has been used widely for large populations. It's very amenable to doing large-scale analyses. And so those aren't particularly problematic. But where you start to run into some of the challenges, at least in our experiences, has been processing all of the untargeted metabolomics data that are generated. And one of the challenges when you talk about processing the data is shown here. So what I'm plotting on this slide is all of the signals are features that might be detected in an untargeted LCMS metabolomics experiment. And each signal or feature is represented as an individual dot in this picture. On average, at least in our experiences, we're detecting 20, 30,000 different peaks or signals in an average experiment. And what happens when you run multiple samples is that we tend to observe drift in the experiment. And when I'm plotting on the y-axis is the amount of drift in one particular dimension of retention time. And you'll notice that throughout the experiment, the drift in retention time can be quite significant, and the drift varies from time to time. It's nonlinear. So here it's in the positive direction, it swings down to the negative direction here, it flattens out a little towards the end of the experiment. And that can really be a problem when we're trying to do experimental comparisons. Now, fortunately, there are a lot of excellent software platforms out there that can help us address these sorts of problems. We often use XCMS, but there are many outstanding programs out there, things like MZMine, MetaboAnalyst, MSDial. Most of the vendors have their own offerings for software processing. But no matter which software program you use, one of the big problems when you try to scale these software up to large cohorts like 20,000 is shown here. And this is a screen capture from us trying to analyze 2009 files with XCMS, so still far short of the 20,000 mark that I mentioned on the previous slide. And you can see the problem here is that the time that it's suggesting this is going to take is over 1,000 hours. And actually, this processing won't even finish. The software will crash before you even get any results. So this is the barrier, and it's one of the major impediments that has limited our ability to do these large-scale population studies. And so what I'd like to talk about is how we can overcome this so we can do large-scale studies, as I've mentioned, in the same spirit that other omics like genomics has been able to accomplish, which are required and really one of the underlying motivations for paradigms such as precision medicine. To appreciate the approach that we've taken, what I'd like to do is take a moment to provide an overview of how we think about the structure of an untargeted metabolomics data set that is acquired by using an LCMS platform. And I'll point out that the diagram that I'm about to show here is really the product of many efforts from many labs all over the world over the last 10 to 15 years. We spent a lot of time on this, developing both software and experimental tools, but many other labs all around the world have made really important seminal contributions to helping establish this perspective that I'm about to show. So the first thing that I'd like to say is that it's important to recognize that in an untargeted LCMS metabolomics data set, you're going to have signals, peaks, or features. I'll use those words as synonyms, but you're going to have signals that are biological and some signals that are non-biological. And what I mean by non-biological is that these signals are not derived from the biological sample. And I'll argue that there are two types of non-biological signals. There are those that are contaminants and those that are artifacts. Sometimes those words are used interchangeably, but we prefer to use them to represent different entities. What I mean by contaminants are that those are signals that represent real chemicals, but those chemicals are not derived from the biological sample. So those could be things like plasticizers from the vials you're using, impurities in the mobile phases, or even carryover from a prior experiment. I do want to highlight that by contaminants, I do not mean environmental exposures. Okay, contaminants are specifically chemicals that are introduced during sample processing that shouldn't be there. Okay, exposures we would still consider to be a biological signal. In addition to contaminants, we also have non-biological signals that we call artifacts. Those do not correspond to real chemicals, but they do cause signals. So this might be things like electronic noise or just small deviations in a chromatographic baseline that your processing software calls a signal or a feature for whatever reason. The biological signals also come in multiple flavors. The first distinction is to recognize that there are those signals that are unique and those signals that are redundant. What I mean by redundant is that one metabolite can show up in an experiment, an LC-MS experiment, as many different types of signals. And the reason for that redundancy, at least some of the main reasons for redundancy, are shown in this diagram. The same metabolite can form multiple adducts. You can form, it can be protonated, it can be sodiated, potassiated, it can form ammonium adducts. There are all these sorts of different adducts. There are also naturally occurring isotopes. 1% of all of the carbon in the universe is C13. That causes mass shifts in chemicals. You can also have oligomers. This is when different chemicals dimerize or trimerize or et cetera with each other. And there's also fragments. So this occurs when a metabolite breaks into smaller pieces at some point during the detection process. Now, among those signals that are unique, you have those that you can identify, which I would classify as your known metabolites. These are going to be things like your glycolytic intermediates, TCA cycled metabolites, amino acids, fatty acids, et cetera. But you also have those signals that you can't identify that I would call novel signals or novel metabolites. So these might correspond to metabolites or lipids that have never been reported before. You can't find them in databases and you can't find them in the literature published previously. So what's interesting is to think about the frequency at which each of these different types of signals occur. And in our experiences, we find that this is a general histogram of the relative distribution of these signals. Now, I want to emphasize that this is the experiences in our lab. But of course, different labs might have different experiences. The amount of contaminants and artifacts is obviously going to be very context dependent. It's also dependent upon the types of samples you analyze. In our lab, we're tending to analyze things like, human blood, as I'm going to focus on in this first part of the talk. But if you're analyzing more exotic samples, maybe extremophile bacteria or other sorts of, peculiar samples, then your distributions, of course, might look very different. But what I'd like to draw your attention to is that the relative frequency of signals that represent unique biological compounds, whether they're known or novel, is a relatively small fraction of the total number of signals that we're measuring in an average untargeted metabolomics experiment. Now, that's not to say that there aren't novel chemicals, and I just want to pause a moment to show some of the novel chemicals that have been reported over the last few years. And this is just really a small sampling of some of those novel chemicals. I don't want to say they don't exist. They certainly exist. I'm just trying to showcase here that their distribution is relatively small to the total number of signals that we're detecting. So these are some of the novel chemicals that have been described recently. The top three here, sulfur-containing compounds, are molecules that we recently reported. This is a collaboration, some work led by Kevin Cho in my lab, but also collaboration with Jim Edwards and Chris Arnett. This molecule here is dimethylsphingosine. It's a version of sphingosine that has these two methyl groups, and it was reported that this is increased in the central nervous system under certain kinds of pathologies. This molecule down here in the bottom left is one that I think is particularly fascinating. This is lactate and phenylalanine, a formation of lactate covalently bound to phenylalanine. This was reported out of Stanford and found that this plays important regulatory roles in processes such as exercise. And then the bottom right is also a really interesting molecule, representative of a whole class, perhaps one of the most exciting areas of metabolomics right now. And these are a bunch of bile acids that are being discovered. What I'm showing you here is a amino acid conjugated to a bile acid. And there's all sorts of different really cool conjugated bile acids and amino acids that have been discovered. And this work is being led by people like Peter Dorestein at UCSD, as well as Andrew Patterson, and many others who are discovering all these cool bile acids. And I think one of the more exciting things that's going to happen over the coming years is trying to sort out what biologically all of these chemicals are doing. But what we'd like to be able to do when we're using these big populations of trying to assess these big populations is take advantage of what we know about the structure of these untargeted metabolomics data sets. So hopefully it will enable us to be able to profile them by still using, again, I want to emphasize using an untargeted type of approach, at least as much as we can. And so the approach that we've been applying is to generate pooled samples. Okay, and the idea here is to take a small aliquot of the samples and mix that small aliquot together to generate a small number of pooled samples. Now, how many pooled samples you create is going to be context dependent. But the idea here is that you create a much, much, much smaller number of pooled samples compared to the total number of samples in your overall experiment. Okay, so hopefully a very finite number of pooled samples. And like I said, even when we're doing these large studies where we're approaching 20,000 samples, we still have relatively small number of pooled samples. And the idea here is that by mixing everything together, the pooled samples can represent the totality of the chemical universe, but do so in a smaller number of specimens. Then what we do is we take those pooled samples, which again is a small number of samples, and we subject them to full annotation. The way that we've been doing that is using some of the software platforms that we've developed. But as I mentioned, there's a lot of different software, technologies that are available, all amenable to this kind of analysis. I'm just highlighting one of the tools that we've developed and we use there, Peak Detective, to annotate all of the signals. But the key here is because you have a much smaller number of samples, you can do complete annotation of those, and the software don't break down because you're trying to analyze really large numbers. And then the idea is that once you've annotated what is biologically relevant, when you do the data processing, you can focus on just those signals that represent biologically relevant peaks. So that would be just the small sliver here, just that small blue sliver that represents real chemicals. Okay, now I want to emphasize that this does not affect how we acquire the data. We still acquire the data in the same way that we normally would, but it just influences how we process the data. And the idea, I sometimes refer to it as the data burden, carrying around all of this noise. These are signals that just don't have biological relevance for the reasons they outlined on the overview of the untargeted metabolomic structure, to get rid of all of that and shed it away. And that really simplifies the data processing so that we can scale things up. Now, that doesn't necessarily correct for the types of drift that I showed you earlier, but we can also take advantage of the pooled samples for those experiments to try to address that. And so what we're doing in my lab is that we're taking the pooled sample and we're running it about every 10 research samples. And then what you can do, this is a bunch of research samples. This is about 2,000 research samples. This is one chemical. LPC 18-1. And what you can see, the x-axis here is the different runs over time, and the y-axis here is the intensity of LPC 18-1. And each of these dots here represents a different run, and the colors represent different batches of about 100. And you can see that over time, the signal intensity of LPC 18-1 does change. So of course, the question is, are those signals changing because of drift, or are those signals changing due to some biological effect? And so what you can do is if you run the pooled sample, you can look at the pooled sample. The pooled sample is exactly the same throughout the entire project. And so the pooled sample should not be changing. And you can use the fact that you are observing changes in the pooled sample to calibrate. We have some machine learning algorithms that we apply to calibrate how much of that change in signal is real and how much of it is due to some sort of analytical variability. What I'd like to do in the closing part of this first section of the talk is describe briefly one large-scale project that we've been applying this approach to, and that is a study that's been funded by the NIA called the Long Life Family Study. And this is actually a project that's been going on for several decades. We've only become involved for the metabolomics component in relatively the last few years. But it's a project to understand healthy aging. And so what this project has done is it's recruited subjects who have a history in their families of having exceptionally long lifespans. So these are families that live to be 100 or more years of age, generation after generation after generation. And then these studies were subjected to all sorts of different kinds of analyses. This is a multi-omics project. I'm just going to focus on the metabolomics today, but we've also collected data for a lot of other types of omics as well. And the subject cohort of this, and I should mention too, that these samples aren't just drawn once from each subject, but drawn several times over a period of many years. And the idea is to perform metabolomics on these. We've got now upwards of 15,000 samples, and we've been working on this for almost 5 years, and we're about 70% done with making the measurement. And what I'd like to do here in the just last couple of minutes of the first portion of this talk is describe some of the results that we've been seeing. And so one of the things that we can do is we can ask if we can find metabolites that are able to predict subjects' age. And I'd like to point out that this work is being led by Paula at the Tufts University, who is an amazingly talented computational biologist who has spearheaded all of the analysis of the data and generated these beautiful plots that I'll show you here today. So there are 308 metabolites that predict age. This is a volcano plot. The y-axis is the p-value, and the x-axis is the full change of those metabolites per year of life. I'm not going to go through all of these here today, but I've highlighted some of the more notable compounds there. And the other interesting thing that we can do with these data is we can ask if metabolites are up or down prior to someone's death, because we're tracking, of course, not only the metabolites, but we're also tracking when the subjects that are participating in the study die. And so we can ask whether or not, what the survival probability is when these metabolites are up or when the metabolites are down. And so on the y-axis here, I'm plotting the survival probability, and this is time after enrollment. And I'm showing you in two different groups, individuals who are less than 90 years of age, who, at least for the purpose of this study, we call it young, and those individuals who are 90 years of age or older. And what you can see, this particular fatty acid, when it's high, you have a higher survival probability than when this is low. And similarly, and that goes for both subjects who are less than 90 as well as subjects who are 90 or older. When it's higher, you have a higher degree of a higher probability of survival. And this is a polyunsaturated fatty acid, or a PUFA. There's been a lot of great reports out there that have shown that PUFAs are beneficial for our health. And so perhaps this isn't too surprising, but it's nice to see that there is a correlation here. And I'll conclude just by showing you one other molecule that we found to be correlated with age, and that is tartaric acid. The relative level here is plotted on the y-axis, and this is a function of age. So each of these dots here represents a different data point that was collected. And you can see the general trend that this molecule is higher in subjects of advanced age. And this is, again, another survival plot, survival probability on the y-axis, time after enrollment on the x-axis for tartaric acid. And what's really interesting, in contrast to the fatty acid that I showed you on the prior slide, what you see here is in both subjects who are less than 90, as well as subjects who are over 90, when tartaric acid is higher, there is a huge drop-off in the survival probability. So you can see that the difference here between the green and the purple, these subjects here, if you're less than 90, you have almost twice as high of a chance of dying if tartaric acid is high. Now, this is obviously just a correlation. We're not suggesting that tartaric acid is doing this, but it is interesting to recognize that tartaric acid is in a lot of health drinks. Tartaric acid is, for example, in these sorts of kumbhaka drinks that are often sold at various grocery stores. And why this is exactly happening, we're not sure. It could very well be that it's not tartaric acid itself, but that the metabolism of tartaric acid is somehow reflective of metabolic health in some way. Nonetheless, we're trying to extend this, and this is going in the, I said the forward and reverse. The first was to make the observation in humans, and now we're trying to do experiments in both cells as well as potentially animals to try to understand what it is that tartaric acid is doing, how it's metabolized. Is this reflective of perhaps maybe some redox balancing with respect to metabolism? Those are the sorts of questions that we hope to sort out by moving towards some more model systems. So that's the first part of the presentation today, representing how we can use metabolomics profiling to study large populations of individuals. But now what I'd like to do is cover the second half of the presentation, and that is thinking about the reverse approaches, where we start by doing metabolomics in model systems, whether it be cells or animals, make interesting mechanistic observations, and then work our way back up to humans, ultimately with validation. And the example that I'm going to present today is studying melanoma. I'm sure most people are probably familiar with melanoma already. It's a deadly form of skin cancer. And we're lucky to have a lot of fantastic clinical collaborators, clinicians that provided us access with a wealth of melanoma patient samples. We also do a lot of melanoma experiments and analyses in mouse models. We have a number of mouse models up and running. But what I'm going to talk about today is using melanoma models, a melanoma model in a model organism, a zebrafish. And I want to emphasize this because I think zebrafish are exceptional model organisms for metabolism and for metabolomics, but I think that currently they're somewhat underutilized. And so I'd like to take a moment here and showcase why I think zebrafish are just phenomenal model organisms for metabolism and metabolomics, and hope to encourage more people to potentially consider using them for their studies. This is a zebrafish there. You can see the scale bars. That red thing at the top, although not accurate with respect to color, is meant to represent a melanoma tumor. The nice thing about zebrafish is they have all of the organs that are required for metabolic physiology. So they have livers, they have brains, muscles, etc. They also have a circulatory system, which is really important because it allows these tissues to communicate with each other and exchange metabolites. And you're going to see that proves to be very important. There are a lot of different well-established disease models for zebrafish: obesity, diabetes, neurodegenerative conditions. What I'm going to focus on here today is the melanoma model. And the way that melanoma model is generated is by putting a mutation that's very common in humans who have melanoma, in BRAF, a V600E mutation. And we do that specifically by putting that BRAF mutation in melanocytes in fish that are P53 null. And then these animals spontaneously develop tumors in their adulthoods. The other benefit of using zebrafish, and we won't get into this too much today, is that we do also perform a lot of isotope tracing analyses to calculate things like metabolic fluxes. To do isotope tracer analyses, you do have to administer stable isotopes. And while you can certainly do that with humans and mouse, it is a little more complicated. It's very easy and convenient to give zebrafish stable isotopes because you simply sprinkle them into the water, much like you would do if you're doing a cell culture experiment. The other benefit of zebrafish is that they are an optimal size for mass spectrometry-based imaging. So this represents a zebrafish that was sectioned, put on what effectively is a microscope slide. Then you can analyze a zebrafish with either DESI or MALDI. This is a image of lactate. You can see that lactate localizes, for example, here around the eye of the animal, and this is the tail at the other side of the animal. So it's really great for doing these whole body studies. And the reason this is proven to be particularly informative, whether you're doing whole body analysis with imaging, or you can also take an individual fish, you can take the serum out, you can take the liver out, you can take the brains out, and you can do analyze each organ from individual fish by LC-MS based on targeted metabolomics. And so what we did is we took the melanoma model. These were animals that had melanoma. And then we performed metabolomics, not just on the tumor tissue, but actually tissues across the entire body. Again, using a combination of both imaging as well as dissecting out the individual organs and analyzing them by LC-MS. And this is maybe one of the more interesting slides that I'll show you today, because what it demonstrates is that when an animal has melanoma, in this case, a tumor, and I'd like to also point out that these results are not specific to zebrafish. We've now replicated these results in other model systems like mouse. But what you'll notice is that there are really dramatic changes across the animal due to the tumor. Okay, so each one of these rows here represents a different metabolite. So I'm showing you just a handful of maybe 50 metabolites or so. And what I'm plotting is that metabolite in the tumor animal compared to that metabolite in the wild type animal in this particular tissue. Okay, so the tumor animal has a melanoma tumor. These tumors have not metastasized, and there's no reason to think that the liver would necessarily be implicated, at least in the phenotypic component of the disease. But you see that there are dramatic changes in the liver. The liver rewires its metabolism in animals that are bearing tumors compared to animals that are wild type. And the same is true of the intestine, the same is true as of the eye, the same is true of the brain and the muscle. And so one of the really interesting questions is, why is it that when you have a tumor that's, at least superficially localized, the pathology is seemingly localized to a very small portion of the animal, how this induces vast metabolic changes across the entire organism? And one of the things that we're really interested in my lab is trying to understand what the origin of all of these metabolic changes are. And what I'm going to do here in the next few minutes is just give you a little bit of a perspective on why, on some of these metabolites, how we've explained at least some of these metabolites, just focusing on a small number. So we know, and from the experimental data, we can determine that these tumors take up a lot of glucose. And that glucose is rapidly metabolized through glycolysis into pyruvate, and then that pyruvate is reduced to produce lactate. This is not surprising. This is a phenomenon known as the Warburg effect that was first observed by Otto Warburg more than 100 years ago. And it's well established that this phenotype occurs in most types of cancers. But what was really interesting was we found that these tumors not only take a lot of glucose and transform it to produce a lot of lactate that gets excreted, but these tumors take a lot of the pyruvate and convert it into alanine. Now, when you go from pyruvate to alanine, that's an aminotransferase reaction, and that requires that nitrogen be added. So you're just adding a nitrogen to pyruvate to make alanine. Then using isotope tracers, we can follow that alanine, and we can determine that the alanine is released from the animal tumor. It enters the bloodstream, it circulates in the blood, is subsequently taken up by the liver, where the liver takes it up and converts it back into pyruvate. And in the process, now when you go from alanine back into pyruvate, we're removing that nitrogen. And that nitrogen then is in the right location, namely the liver, where it can be used and excreted as waste in the form of ammonium. And one of the nice things about fish is that you can sample the water, and by sampling the water, you can determine what the animals are excreting. And what I'm plotting here is an analysis that we did using a protocol that we got from Marcia Hagis's lab out of Harvard to measure ammonium, and this is micromolar. And that's the amount of ammonium that's in wild-type animals compared to the amount of ammonium that's in the water from cancer subjects. And you can see that the amount of ammonium goes up more than twofold in these subjects. In fact, these animals that have tumors excrete so much ammonium, it actually causes problems for us. We have a zebrafish facility in our lab, and it causes problems in regulating the conditions of the tank because there's so much ammonium that screws up the way that we're filtering the water, buffering the water. Now, what happens to that pyruvate that's present in the liver? Well, that pyruvate can go back up to produce glucose. That's a process that's essentially the opposite of what you see in the tumor. So instead of that being glycolysis, that's a pathway called gluconeogenesis, where you take pyruvate and reconvert it into glucose in the liver. And that glucose is then excreted from the liver, enters the blood, and travels back to the tumor, where it is taken back up and the cycle starts again. So what I'm describing here is a tumor liver cycle, and it represents what's happening, that the tumor is taking up nutrients, turning it into waste, excreting the waste, and then essentially co-opting liver metabolism to convert that waste that the tumor has excreted back into useful nutrient that goes back to the tumor. So it's essentially co-opting the liver to convert waste, tumor waste, back into tumor fuel. Now, gluconeogenesis is a process that all of our bodies are capable of, and depending on what time of the day it is and when the last time you ate when you're watching this seminar, you might have gluconeogenesis going on in your livers. You know, when you go long periods of time without eating, your body tries to maintain glucose levels in the circulation, and one of the ways that that is accomplished is through gluconeogenesis. But what's important to point out in these animals that have melanoma tumors is that gluconeogenesis is activated all of the time. It's constitutively active even after these animals have eaten. One of the questions that we had is, where does that nitrogen come from? Why are these tumors putting all the nitrogen on pyruvate? Where does it come from? And what we found is that these tumors are addicted to branch chain amino acids, and that is the source of the nitrogen. We did a lot of tracing, putting a lot of different N15 on a lot of different potential nitrogen sources, and found that the majority of the nitrogen is coming from branch chain amino acids. And this is apparent if you look at the expression of an enzyme that's required to metabolize branch chain amino acids, BCAT1, and you can see that that enzyme, that gene is very highly activated in melanoma. It's essentially off in healthy melanocytes. And we see the same exact thing when we look at human subjects that have melanoma, indicating that this pattern that I've shown you is not limited to zebrafish, but also might be occurring in human people as well. So the question is knowing this cycle, can we potentially block liver metabolism? And by modifying liver metabolism, is that sufficient to prevent these tumors from growing? Can we actually target tumor metabolism? You know, can we develop anti-cancer therapies by targeting the liver? Which is a little strange. We tend to think of anti-cancer therapies, we tend to think of as going after the tumor. But we did that, developed a way to do that using an alt-I drug that targets and blocks that process in the liver. And you can see that after just 10 days, we do see a substantial reduction in the size of the tumor in animals that are receiving that drug compared to the healthy controls. Now, I do want to just take a moment and say, remember that the idea here is going reverse. So starting with model systems, working our way back up to humans. We're in the process of doing that. And I also want to point out something that I think is really interesting from a multi-omics perspective. When we do metabolomics and you're trying to look at something like glycolysis and gluconeogenesis, those are different pathways. Glycolysis is glucose to pyruvate. Gluconeogenesis is what's happening in the liver, pyruvate to glucose. Okay, but what's important is that all of the metabolic intermediates are the same. It's the same pathway in both, whether it's glycolysis or gluconeogenesis, the metabolic intermediates are the same. But what is different is some of the enzymatic machinery. So just looking at pool sizes of metabolites really doesn't help us very much in trying to differentiate between glycolysis and gluconeogenesis. We really need isotopes to do that. But if we look at the enzymatic machinery and do targeted proteomics analysis on those enzymes, then that provides us perhaps a little more insight into which of those pathways is active. And so when we look at FBPase here, which is utilized in gluconeogenesis, you can see that it's higher in the cancer tissues, livers from mice relative to control mice. And we see the same patterns in human subjects when we look at human patients, the livers from human patients, that this phosphatase is increased in cancer relative to control. Now, I do want to point out that we ultimately are in the process of trying to validate this in humans. And we are, although I think the protein analysis that I just showed you provides a good indication along with BCAT1, we do want to validate this in humans. So we're in the process of doing this on a clinical cohort. There's a couple different ways that you can do this. You can infuse C13 glucose, which is shown here. But alternatively, another way that you can do this, and that requires, of course, setting up the patient with, setting up in an IV and infusing them usually before the tumors are resected. But another way of doing this that at least for us has proven to be a little more practical and perhaps a little easier because you could do it outside of a clinical setting is to instead of using C13 glucose, to do these experiments by using deuterated water, D2O. And the way we've been doing these experiments, and this is shown here in this recent paper down here that was just we recently published in Cell Metabolism, is that we're giving these subjects deuterated water for about 5 weeks, twice a day for 4 times a day for a 5-day prime period, and then twice a day for this 4-week follow-up, which gives us a good enrichment and allows us to assess these fluxes without having to do the infusion in the clinic, like I'm showing you on the left-hand side of the slide. So with that, I'd like to just make a couple of concluding comments before we wrap. And one thing that I'd like to point out is that what we're seeing in the studies that I've shown you today, as well as many other studies, is that all of the cells or many of the cells in an organism are metabolically coupled to one another. They form metabolic partnerships where the output of a particular tissue or a particular cell is the metabolic input of another tissue and another cell. And we saw that relationship here today when we thought about the tumor in the liver. And I don't think this is just limited to healthy tissues, but I think it's going to occur in a lot of different types of tissues and pathological tissues as well. So if cells are all metabolically coupled, it raises the question of what happens when one cell or one tissue becomes pathological. And you can imagine, as I've shown you here today in the case of melanoma, that even though it's just one tissue whose metabolism becomes pathological, that can initiate a cascade because everything is connected, that can initiate a cascade that leads to perturbations that are really system-wide across the organism or across the human. And this occurs not only in other tissues, but also by looking at circulating metabolites that are in the blood. And I think, at least in my perspective, this has provided kind of a renewed sense of enthusiasm for how we think about doing blood metabolomics on samples like plasma and serum. And what I've shown you here today is that tumors are interacting with other types of tissues, and that's really interesting and potentially opens up some novel therapeutic paradigms, because instead of, as we have historically, primarily focusing on developing therapeutics that target the cancer, it might be possible that we can develop therapeutics that target what are recognized as otherwise healthy tissues, but who are intimately involved in the metabolic processes of what's happening in the disease site here in the tumor. And like I said, I don't think this paradigm is going to be specific to cancer. I think we're going to see this occurring in a lot of different disease states. So with that, I'd like to thank the folks in my lab who are responsible for this work. Some of them are pictured here. I'd like to particularly thank Ethan, Mickey, Kevin, Miriam, who contributed to that, and Nate, who contributed to the first part of the talk, provided, like I said, it's been a very long project. I can't name everybody who's been involved in that, but they've really spearheaded some of the work that I've shown you there to try to scale up metabolomics and trying to tackle what is initially seemed very overwhelming to try to profile almost 20,000 people. And then I'd also like to thank Maddie Jackstadt and Fuad. They were the leaders of all of the zebrafish work that I showed you. I provided the publication at the bottom left-hand side of the slide there, but they led all of that work from developing how to establish a workflow for doing zebrafish metabolomics to the isotope tracing to interpreting all of these results. So with that, I'd like to thank you for your time. I'd be happy to entertain some questions if time remains. Thank you.
Thank you very much, Professor Petty. It was really, really nice talk, especially how we connected the first part with the large population studies to the models. It was really, really elegant. Yeah. So now we will take the questions and answers session. So please, I advise all the attendees who are interested in something, please write your question into the Q&A box, and then it will be answered. So we have lots and lots of questions, so I have to go through. We will just give, I don't know, 15 minutes, for example, or something that you have time to answer. And there's one question that connects also to one question I was about to ask. And that is concerning your long life family study.
Mm-hmm.
And as this is really a big study concerning thousands of samples, and we are also doing something similar, I was interested, what is the most challenging part of conducting such a study, like from the collection, storage, QCs, analysis, data processing? What is the most challenging part, probably? And maybe if you have some tips for people that are about to conduct such a study from your experience.
Yeah, no, it's thank you. Wonderful question. So first, I just want to pay some credit to Mike Province, who leads this study, had been leading it for many years, and who is an amazing leader for the project. I think these large scale studies are really tricky because once you start, you really can't change. So, like I said, we've been acquiring data for this project for 5 years. And if you go back and say, 5 years into the project, if you say, oh gosh, I don't really like this helic method, we should have, so-and-so just published this really cool gradient and I want to use that. The thing is, you can't go back because the data, once you start the study, it's really irreversible. It's not like when you have a cohort of 20 and you can say, oh, here's a new method, I'm going to go try it on my 20 samples. So I think that, and I would argue this is the case for all untargeted metabolomic studies, but particularly these large-scale studies, that it's absolutely essential that you spend as much time on the front end deliberating about every single detail that you could possibly think through, and ensure that you're really comfortable and satisfied with how the protocol works, how the workflow is set up, how the processing, because it's not something that you want to, as the study goes, say, well, I'll figure out how we're going to do data processing later, or I'll figure out where the best place to get the column is. The other thing I would say, we made this mistake, and because some of this was going on during COVID, is make sure you have tons and tons of supplies. You don't want to start a large study like this and only have 2 columns, because we ran into the issue during COVID where, as many other people, supply chain issues where certain things were suddenly not available, and we're in the middle of our runs, and what do we do if we can't get this exact column? You can't just switch columns in the middle of this large scale study. We were fortunate to be able to get the columns that we needed, but making sure that you have all the supplies, you've deliberated as much as you possibly can about the workflow, It's also helpful, although I realize this isn't always practical, to have sibling instruments so that if one instrument goes down, we have 3 instruments that are exactly the same. We're doing all this on Agilent QTOFs. And that way, if something goes down, we have other instruments that we can pivot to so that we're not stuck, waiting for service. So there's some factors, but, I think that they're very complicated studies, and I wish anybody embarking on this. I think it's really exciting, and I wish everybody luck who is taking on this sort of effort.
Thank you. There's also like a follow-up question to that from the audience. I will read it whole. It's from Daisy Zhao. So thanks for the informative talk. We are also working on clinical metabolomics for a long time. For long-term prospective cohort study, samples collected at the beginning of the study, can they be degraded after years when the study is completed or during the study? How can you ensure the analysis of results, for example, of the outdated samples is reliable? And if you have some suggestions for the collections of the studies, of the samples that are collected for a couple of years. So
yeah,
that's it.
Oh, excellent question. So there's been a lot of studies led by others that I don't have the references handy, but if the person asking the question wants to email, I can pass them along. But there's been some really nice studies where people have looked at sample stability. So I'll point you. And I think the Metabolomics Society had some working groups as well that has investigated that. So I can help with find some of those. There's some really great stuff out there already. But one of the details that I didn't include. So you're absolutely right. You do worry about sample degradation. One area that we found is particularly sensitive to that is redox active metabolites. So for example, if you're trying to look at things like NADH or NAD plus or NADPH and NADP plus, those are particularly sensitive to storage conditions, and you do see that those redox ratios can change if the samples aren't measured appropriately. But one of the details that I left out that we also do, and I appreciate the question, and maybe I should think about including this, is that in addition to running the pooled samples, we also run a reference material sample. So we're using the NIST 1950 plasma that we also run. And that way it does give us, we have more of a ground truth, so to speak, because that is a sample that, you can purchase. So you can, try to keep authenticity in that way, because you can always get new material. But there's a ton of data out there on it. And so we also intercalate within our experiment running some of those reference materials so that we have not only the pooled samples that we can use to monitor what's happening with batch and time, but also those reference materials. And you can put those reference material specimens through the same workflow as well at the same time. So we use 96 well plates, and we sacrifice a lot of real estate on our plate to cover these pooled samples and the reference material. So it does decrease your efficiency. And as you're sitting there pulling your hair out, trying to scavenge money to support this, you really wish you could take those wells on the 96 well plate and use them for the research samples. But we find it's really productive if you can sacrifice 15% of the wells to have these replicates, that it goes a long way. So that would be my advice there, I think, is to think about using something like the NIST 1950 platform.
Okay, thanks a lot. Also, as we are talking about this quality control, there are 2 more questions kind of connected, so I will try to interpret this. Basically, they are asking how many pool QC samples would you use for 2,000 sample project, or for example, the 15,000 sample project? What are the considerations?
Yeah, no, it's a wonderful question. Kind of the $1 million question that I left off and just sort of magically waved my hand and said, we'll use pooled samples. The other thing is that you can also use internal standards here. I didn't mention that, but we have done some of these projects where we use Cambridge Isotope Labs to purchase kits of isotopes that you can spike in. And that's another way. You can spike them in both the research samples as well as the reference materials, the quality control samples, to try to get insight into this. So in our experience, in terms of how many samples, how many pooled samples we're using, we don't have a definitive ratio. I wish I could tell you, you know, it's 10 to 1 or 100 to 1, but we don't have a definitive ratio. It's been very contextual. And part of the problem has been, and maybe you're facing these challenges as well. Part of the problem has been is that at the beginning of a study, you don't actually always have the samples. So the LFS study that I outlined here, it's a prospective study. So every period of time, every couple of years, we're getting whole new batches of new blood draws from new people who haven't yet, or not from new people, but we already had previous samples from them, but now we're getting new samples from them from those who are still alive. And so there's no way, of course, if the study is 5 years and you're going to get, half the samples or, another 5,000 samples in the last 2 years of the study, you can't make a pooled sample that you use on year 1. And so that's actually caused us to have more pooled samples than we probably would have liked for this particular study, because as the new samples are coming in, we're having to make more pooled samples. But this, again, I think really highlights the value of using the NIST 1950 because and this is awesome too, because we're doing plasma, and that is a plasma sample. I think it gets much harder if you were to do something like that, that for which there's not a good reference material available. We've struggled with that a little more. If you can't go out and buy a 1950 plasma, what do you do? But for us, yeah, our numbers have been a little higher than we'd like because of this prospective enrollment in that case. But, we're still looking at, even for such a large study, still trying to keep those numbers under 100.
Okay, thanks a lot. That was very informative, and I think many people can use that in their own design of experiment and such studies. We have a few other questions similar together, so I will try to read them together somehow. It's about the nice plot you showed about the lysophosphatidylcholine 18, the retention time drift. And basically, just a question: if this particular shift would be considered like a good metabolite lipid, like a good performance, and how did you did the corrections for it?
Yeah, no, that's a great question. So I apologize, a little bit of confusion there. So on the very first slide, when I showed drift, I showed the drift in the context of retention time. That's what I showed on the first slide. But when I showed the LPC, what I was actually showing there is a change in intensity. So that's the intensity of the molecule. And you can imagine if you take the exact same sample and you run it for a year straight, although we'd love to see the exact same intensity, right, for all the year, of course, as the fall and the winter and the summer, and depending on where you live, the humidity and everything changes, you get these patterns where the intensity drops and can come back up. You get these oscillations. And so what I was plotting there is the intensity. And that's really important, right? Because for sure, you're going to get dramatic changes in the intensity of signals over 5 years on your mass spectrometer. And those changes can actually be quite large, and you don't want them to mask the changes in the biology, because if you get a change, 50% change in the metabolite, if the drift in the signal is bigger than that, then that's not useful. So we actually, the way we do this is we have about 10 different machine learning algorithms that we test in every study. And interestingly, and somewhat surprisingly, actually, we don't always find that the same machine learning algorithm works the best from study to study. And that probably has to do with the instruments we're using, the design of the study. For this particular LFS study, we're using a random forest model. Is the model we've been using. But like I said, for whatever reason, we don't always find they're the best. So that is the best. And so if that person wants to email me, I can send them how we do this, and it's been published. We have a Cell Reports Medicine paper that came out a couple of years ago that describes how we do this. But for every time we run one of these cohorts, we test all of those and then look for which performs best, and then use that for the overall sample, the whole entirety of the sample analysis.
Okay, thanks a lot, and also thank you for being so open. So I can let the attendees know that if they are very interested about something and it's not completely answered here, and they are still wondering, so they can email Professor Patti directly.
Yeah, absolutely.
If I understand correctly.
Yeah, absolutely. I'll try
Okay.
to I'll try to answer as many as I can.
Okay, thank you so much. There's also a few questions connected to the survival plots, as you showed, the survival curves. And one question is concerning why did you choose the 90 cutoff? It was, I believe, for the age, or if it was, and also how is it calculated? But probably, yeah.
Yeah. So those plots were created by Paula, who's a computational biologist at Tufts, and one of the reasons that we used 90, of course, you could use it. I think what we really did there is look at the distribution of the ages of the people in our study, and also when people are younger, the probability of survival is much higher. And so what we're doing there is we're essentially looking at how many subjects at a given age died, and then we measured we measure a particular metabolite. Did that metabolite change prior to their death relative to their measurements that we did previously? So you could imagine, that glucose is normal, and then suddenly glucose plummets really, really low, and then the subject dies, that year. And so what we're trying to see is if a change in a metabolite would predict whether or not an individual is likely to survive or likely to die. Now, this is obviously totally correlative. So we cannot 100% cannot say that metabolite changed and that caused the death, right? And this gets into the forward direction of studying human metabolism. These are only phenomenological observations and should not be interpreted as causal. And you could also imagine that when somebody gets really sick, if somebody gets cancer or somebody gets, really severe, whatever it may be, COVID, and subsequently dies, a lot of metabolites are going to change, right? And it's not to say that metabolite was keeping them alive or that metabolite changing was causing their death. So these are, I think, really complicated plots. I presented them maybe a little too superficially in terms of how much, biological insight they can have, but nonetheless, I think it is really interesting. And we try to use that 90 cutoff simply based on the age distribution of the people that we had, and then when they are dying. Because obviously, if people aren't dying, then you don't get a very interesting, you just get a flat curve across the top. So, because then you have 100% survival, irrespective of whether or not the metabolite is changing.
Okay, thank you a lot. The questions are pouring in, so I will just ask you if you have time for like 2-3 more.
Sure.
Maybe.
Yeah.
Oh, okay, perfect. So we will continue, and now I will switch my words to Daniel, who can read some other questions.
Thank you, Aleš. There's a question coming from Greg. He says, they say, thank you, Gary, for this informative talk. There was a number of observations on established phenomena, e.g. Cahill cycle and Warburg. How does metabolomics address the missing or unknown biology, or more targeted approaches required to address such a concern?
Yeah, it's a great question. So I think the question there is, we two phenomena mentioned, the Warburg effect and core recycling, both processes that are fond to my heart. The question, I think, is how insightful is targeted and untargeted approaches for investigating those phenomena? It's really a great question. I think both targeted and untargeted metabolomics are useful. And it really is a matter of preference. I think it just kind of depends on what your questions are. Maybe what the question's insinuating is that if you want to think about the Warburg effect, does it make sense to do untargeted profiling, or would it maybe make more sense to do a targeted analysis just of glycolysis? And you could certainly make that argument. In this particular study, we didn't go in with a particular hypothesis in mind. So we started with the untargeted analysis because we didn't go in saying, for example, is Warburg altered due to melanoma, or is Warburg different in melanoma compared to whatever? We didn't go in with that hypothesis. We did untargeted, and it just turned out that the data that were altered, we were able to interpret in the context of core recycling and the Warburg effect. But I think you're absolutely right that ultimately, if you do want to transfer, I think untargeted metabolomics, this again is in the spirit of starting forward and kind of working your way along. you tend to start, at least in our lab, we tend to start with untargeted metabolomics, come up with some kind of interesting observation. And then usually that motivates more targeted studies. And often, in many cases, those targeted studies are isotope tracing, where we're, in many ways, an isotope tracing experiment is a targeted analysis. You're picking a certain nutrient that you isotopically label, you give it to a biological system, but you have to pick a nutrient, glucose, glutamine, lactate, whatever it may be. So in some sense, when you're doing that, you're already doing a targeted analysis, and then you can calculate fluxes of particular pathways. That's also a targeted analysis. So usually for us, it starts with an untargeted, it generates a hypothesis, and then we go in and further investigate that hypothesis using both a combination of untargeted technologies as well as targeted metabolomics and targeted tracing methodologies. I hope that answers the question.
Yes, thank you for the expansive explanation. And I also have another biological question coming from Otta. They say, thank you, Gary. Can you comment on the relative size of tumor in the zebrafish model in context of the average size? But in the fasting human, this alanine contribution to gluconeogenesis is significant?
Yeah, it's a great question. So I think what this individual is asking is are the tumors proportionally, like you can imagine so first, let me take a step back. The question I think that is being raised, and it's an excellent question, is does the impact of the tumor, does the size of the tumor impact the systemic effects that it has on the rest of whole body metabolism? You can imagine that if a tumor is really, really tiny, does it still lead to systemic alterations, or does it have to be really, really big to cause all of these alterations that I showed? At least what we found is that the size of the tumor is important until you cross a certain threshold. So when the tumors are relatively small, like thousands of cells, they seem not to have this impact. But then at a given size, they seem to somehow start behaving like a physiological tissue, start acting like they get integrated into the physiology of the host, and then they start interacting with other tissues. And then after that happens, that seems to be relatively constant as the tumor gets bigger and bigger and bigger. So it seems to be kind of a binary activation rather than kind of a gradual shift in metabolism over time. To directly answer the question, though, about the size, the picture that I showed of the fish with the red tumor is approximately to scale. That would be a large tumor on the animals, but some of the tumors are much smaller than that. And in the study that I showed, we didn't just use big tumors. We used a combination of big tumors and small tumors. So it's really an average of tumors. But we are in the process of doing subsequent studies, and this work is being led by another person in the lab, Moongu, with tag teaming it with Maddie, that we're actually comparing sizes and trying to sort some of this out more specifically. But I think that's a really good question. Hopefully that answers your main inquiry.
Aleš, are you there?
Yes. Okay. So do you still have time for like 2 more questions, or
sure,
what is
let's
your?
do 2 more.
Okay, that sounds reasonable. Okay. I had this one again. Some of there. There's a question about the second part of the presentation. What is the infusion strategy for the deuterated water, and how is it beneficial compared to C13 glucose? Like infusing
Sure, yeah.
directly.
I'm going to stop sharing my screen just while we do this. Of course. Did that work? You're not seeing am I now bigger?
Yes, it's perfect.
Okay, perfect. So yeah, so that's a great question. The deuterated water actually we're giving that to them orally. So it's not that we're infusing it. We're giving it to them And they come in these little tubes. So what happens is that we give them little small, what effectively look like Eppendorf tubes. They're of course sterilized, and they have a small amount of the D2O in it. And then the person is to take them, as according to the schematic that I showed. And when they take them, the clinical coordinator, then when they come in to see the project coordinator or the nurse, they bring the empty vials with them. And that's one way in which we monitor to ensure that they've actually consumed the water. We also do blood draws throughout the study, as well as saliva collection to ensure that we're seeing the appropriate D2O there. And if there's any snafus where we're not, they don't bring back the right number of vials, or we're not seeing deuterium in the saliva as we go, then we know that there's a problem there, and we flag it. But it's not an infusion. It's much easier because you could just drink it. And we're getting really good enrichment and been able to calculate some really cool things doing that way, which is much easier, obviously, than having to hook up an IV and do the infusion directly before surgery. It just simplifies things logistically.
Okay, thank you so much. I would just like to tell the attendees that we will read the last question because Professor Patty's time is sacred. We don't have to keep him too long. So we will have one last question, and the rest of the questions, if you are still interested into something, you can directly address Gary. We shared his email, like the official one, through the invitation. So you can do that, and we will read the last questions. There are actually a few questions at the same topic again, so that's nice. And it is about confounding factors. So I think it's maybe aiming to the long life family study, and probably also any other type of study where you deal with human samples. How do you treat these confounding factors? How do you work with sex, ethnicity, smoke habits, and maybe even medication?
Yeah, that's a great point. I think this is actually why you need large numbers. So of course, when you do mice, you can put them all on the same diet, put them in the exact same room. It's the same temperature. You can regulate everything, so they try to keep them as harmonized as possible. But when you do people, that's not practical. And all these things that are referred to as confounding factors are actually what makes us individuals. People have different exercise habits. People are exposed to different kinds of environments. People eat different diets. They're experiencing different levels of stress, et cetera, et cetera. And this is all part of the individuality. And I would say part of the individualized or personalized medicine or precision medicine. The idea being that each of the subjects who have a confounding factor might also have a different disease process or different disease progression or require different therapeutic intervention because they are distinct. And so what we're trying to do is not necessarily recognize them as say, confounding factors, but trying to have a big enough sample size that we have enough smokers and non-smokers so that we can actually try to evaluate the effect of smoking, or sex, males and females, or age, or drug treatment. So the idea is to try to have, and that's why I think that's really the value in doing these large population scale studies, is that a lot of the things, if you only have 10 people, the likelihood of having enough statistics to average that stuff out, it's just never going to happen. But when you go to 20,000, suddenly you start to get more statistical support for trying to evaluate these trends that might be small, but when you have big numbers, they become large enough that hopefully we can assess them. At least that's our aspiration. It also helps that we're not just acquiring metabolomics data, we're also acquiring all of these other omics, genomics, proteomics, all kinds of health information, as well as phenotypic assays like grip strength, gait analysis, all these other types of information as well. So we not only have we're not just with the mass spectrometry data, we have all of these other profiles as well as all this other information. And one critical question that you might ask, and I don't have an easy answer for this, is, well, how do you take all of this and put it together? What's the workflow to take all of this vast amount of information and all these variations and give us an answer that tells us something interesting? And that's, I think, really hard, but was mentioned in the introduction. Recently, the Multi-Omics of Health and Disease Consortium was initiated by the NIH, which is a big effort to try to collect omics data, similar to what I showed you for LFFs, but for several different different kinds of diseases, 6 different diseases. And one of the charges of that consortium with the NIH is to try to develop better solutions to address these large scale populations and multi-omics integration and the sorts of confounding factors that you've mentioned more rigorously from a computational perspective.
Okay, thank you so much. So yeah, this was our last question. Thank you, Professor Petty, so much for your time. Just for your information, we had over 200 attendees, and now it's still during the Q&A section, there are over 80, so it's very nice, according to our numbers. There are still lots of questions, so people, if you really think that it's important and you are still interested, you can write an email directly to Professor Petty. And if you have any other questions, just directly to us, the Metabolomics Society and the Early Career Network. Thank you so much for your participation. Thank you once again, Professor Petty, for your time, and really excellent talk and spending your time at all the questions really deeply. Yeah, thank you so much. And lastly, of course, to my co-host, Danielle. So thank you so much, and have a great day.
Have a great day, everyone. Bye-bye.
Bye-bye. Thank you.